Well, hello everybody. I think I am live. It's great to be here. I'm coming to you from a beautiful sunny morning in Seattle, if you can believe it. My name is Stephen Smith. I am at Seattle Children's Research Institute and also the University of Washington. And I'm going to be talking today about decoding CSF mutant Huntingtin. So we use a technique called IPFCM, or immunoprecipitation flow cytometry. And in IPFCM, we use 5-micron microbeads coupled to capture antibodies. And these allow us to capture protein out of a solution. In this case, we're capturing mutant Huntingtin protein. We then use fluorophore-coupled probe antibodies that also bind to a different spot on the same protein. And we run these beads through a flow cytometer. And the output of the flow cytometer is a histogram like what you're seeing on the right side of the slide. And this reports the median fluorescent intensity of all of the beads that have passed through the flow cytometer. So IPFCM is basically, you can think of it as an ELISA on a flow cytometry bead. It's a very sensitive way to measure proteins. Now, my lab has specialized in using IPFCM for various reasons. One example here on the right is we can detect protein-protein interactions. This graph on the right shows us detecting a physical protein-protein interaction between the T cell receptor, CD3, and a co-associated protein, ZAP70. And you can see how as we put in fewer and fewer cells into our reaction, the signal goes down. But you can see if you compare to the Western blot next door there, that we can be orders of magnitude more sensitive than a Western blot when we use IPFCM. A second sort of modern relevant thing that we've used IPFCM for is we coated our 5-micron beads with the spike protein from SARS, and we were able to then incubate these beads with serum from post-convalescent patients, and we could measure antibodies to COVID-19 spike protein in human serum. So the reason that we use IPFCM for all of this is because it's highly sensitive. We've published that we can detect T cell receptor from as few as 300 T cells. So it's quite sensitive. Our flow cytometer has a large dynamic range, and the signal is linear throughout most of that dynamic range. So it gives us a pretty wide concentration gradient that we can measure. The readout is inherently quantitative in the form of the median fluorescent intensity, and we get repeated measures. We measure beads, we measure thousands of beads per experiment. So rather than making a single measurement in a Western blot, you make repeated measures on beads, and it gives you a little bit more confidence in your measurement. So our goal here is to measure mutant Huntingtin protein using IPFCM. And what we need to do here on the left is we need to identify antibodies that can bind two different antibodies that can simultaneously bind Huntingtin protein. So if the Huntingtin protein has some sort of weird conformation in the middle that occludes an antibody binding site, those two antibodies won't work in combination. Similarly, if the binding sites are too close together, like on the right, the two antibodies won't work in combination. So the first thing that we had to do to measure mutant Huntingtin was to screen all of the antibodies that we could get our hands on. So we did, I think these are 13 commercially available antibodies, and we used them each as an IP on the left and as a probe in the columns here. And we identified one combination of antibodies that gave us the strongest signal. And this combination was an immunoprecipitation with HDB4E10 and probing with MW1. So that's the antibody pair that we went ahead with. There it is. All right, so this combination is sensitive and specific to mutant Huntingtin. So here we have brain lysate from a mouse that has a human Huntingtin with 97 repeats in it. And you can see we generate a signal. if we use a non-expanded Huntington protein, a mouse that has the endogenous mouse Huntington protein knocked out, or a wild type mouse, here we get a baseline signal. So this shows that our assay is specific for expanded mutant Huntington. We could dilute down this brain lysate 1 to 150,000, and we could still get a signal. So this gave us some idea that we were developing quite a sensitive assay, which of course we would need to reach our goal of measuring Huntington in the CSF. And finally, we measured Huntington in the CSF, and we were able to show that in HTT manifest patients. All right. So the next question we wanted to ask is, can we relate the MFI, the fluorescent intensity of our readout, to the actual concentration of mutant Huntington? And to do this, right, we would just need to generate a standard curve where we had known concentrations of mutant Huntington, and then we would be able to relate that to the median fluorescent intensity of our measurement using some sort of equation. But here we ran into an issue, and that issue is that MW1 binds to the poly-Q region in mutant Huntington, right? And we know from prior work out of Pam Bjorkman's lab with the crystal structure of MW1 here bound to a poly-Q peptide shown in a sort of a crystal, I'm not sure what that structure is called, the line structure in the middle. So we know that MW1 binds to 7 Qs. So depending on the length of the expansion of the mutant Huntington, you could get different intensities of fluorescent signals, right? So here we have a bead, which is bound to a very large mutant Huntington expansion. We see, we have 4 antibodies bound. But we can't really tell the difference between this situation where we have a shorter mutant Huntington stretch, but there is more protein bound. So it's not a simple, straightforward calculation to go from concentration to fluorescent intensity. Instead, what we're going to need to do is to relate the concentration to fluorescent intensity for many different lengths of poly-Q. And to get to every possible length is really a non-trivial thing to do. And with that, I'm going to hand it off to Doug, who is going to talk about the next portion of this, which is trying to make those relationships.
Okay, hey, Doug Langbehn from the University of Iowa. Follow up on Dr. Smith's comments, especially his last couple slides. I will go into a little more detail about this. My first slide here presents a maxim that I constantly need to keep in the front of my mind, and I frequently repeat. The fact that we can measure something doesn't mean that we can measure it well. And the corollary to that is measuring something well depends completely on the context that we need the measurement for. So we want to keep that in mind as we'll talk about some of these issues of trying to quantify these femtomolar concentrations of mutant Huntington in the CSF. So Dr. Southwell shared data with me from 3 different controlled experiments where the control was both a known concentration of mutant Huntington and also known polyglutamine length, Q length within the samples. So this is very similar to what Steve had showed a couple of slides ago. Shape is slightly different here because on the horizontal axis I have concentration instead of log concentration, and the y-axis is the mean fluorescence that comes out of the assay. So I've plotted all 3 of these to the same scale, and there are a couple of kind of semi-quantitative features that we can, I think, immediately see are consistent among these 3 assays. First of all, the fluorescence is going up with increase in mutant huntingtin, so that's reassuring. But again, it's very obvious here. The numbers are the Q lengths, known Q lengths of these control samples, and it's obvious that the amount of fluorescence depends pretty heavily also on the Q length once we start getting into the range that is relevant for Huntington's disease. And we can also see that when we get into longer Q lengths, that ideal linear relationship between concentration and fluorescence actually no longer is quite linear, and instead the curves start to, well, they turn into curves. They're curving downward, they're concave. So we need to worry a little bit about that feature of things as well. Now, there's also an obvious inconsistency when I look at the plots of these three assays, again on the same scale, and that is the fact that the actual numbers we're getting from different combinations of Q lengths and concentrations are different on these three experiments. So, for example, if I fit a calibration equation based on experiment 1, it actually would not work very well on experiment 2, experiment 3, etc. And so there lies a practical problem with all of this. So what's the source of this variability? What do we need to do about it? Well, the first thing we typically look at when we're trying to evaluate the quantitative technical properties of an assay is we look at what's called technical replicate intercorrelation or reliability. So plots on the previous page were actually the means of 3 repeated measurements of each sample. They're triple replicates, 3 different aliquots. They get measured separately, but during the same assay. You hope they get pretty much the same answer, and indeed they do in this case. The intercorrelation is excellent for all 3 of these experiments above 0.9. So we know that there's no issue there, and that's very reassuring. But it's a little difficult to know where to go from there to try to sort out, if we do need to sort out what else is going on with variability in these assays. I think obviously there can be some unknown variables in the assay conditions that influence calibration, and talking to Dr. Southwell a little bit about this, I think there are actually some known but very difficult to control variables within the assay that are probably influencing the calibration. There are issues with potentially different combinations of protein species and fragmentation and so forth that may possibly vary by sample. And this, to some extent is biological, but also could be an artifact. It could be one of the artifacts that can come up from inconsistent handling, processing of samples, and there's always stuff that there are always more potential problems in that area than we understand. So it's a little unclear what's going on. But in any case, it does look like that within a given assay, we can come up with a calibration that works well for that assay. And based on those 3 experiments, the formula at the bottom of this page seems to be a general form that works very well for calibration from assay to assay. The plot on the right, the scatter plot, is the calibration fit line versus log concentrations and log, or the prediction from log concentrations and log fluorescence from one of the assays. So you can see that there's a good fit there. But 3 constants in this equation, the K and the Y and the X, are all things that need to be estimated statistically. And to do that, we would need something very similar to one of these experiments that I showed on the first slide as just a calibration that's part of an individual assay run. Gets kind of complicated and kind of a big task because of the multiplicity of probably needing to look at, maybe 4 to 6 Q lengths rather than just calibration of a single concentration, which is usually what's done to calibrate assays where we don't have to worry about different size molecules. So to get absolute quantification, or a good approximation of absolute quantification, it's clear that we do need to know these polyglutamine lengths, these Q lengths. And that immediately brings us to another challenge when we think about mutant huntingtin coming from the brain in the CSF. We know about this phenomenon of somatic expansion, whereby the polyglutamine lengths are increasing in the brain and increasing actually kind of inconsistently. We know that we don't wind up with a single Q length in the proteins that we're trying to measure, but rather a distribution of Q lengths within the CSF protein. So it's not as simple as just knowing the CAG length from a white blood cell and the concomitant Q length that comes from that CAG length. A lot we want to know, we don't know about what else is leading to this distribution of somatic expansion in the brain. But I think the immediately practical point is that it's my understanding there's no practical way of actually measuring that distribution of Q lengths. I think it hopefully would be satisfactory if we could even get kind of a mean Q length out of a CSF sample. But again, as far as I know, there's presently no method available to do that. So this brings up just one last idea I wanted to touch on quickly. It may be for some applications we don't actually need an accurate absolute quantification. Relative quantification for, say, a given individual over time might be adequate. If the application is a longitudinal follow-up study, i.e. a clinical trial, it might be sufficient just to be able to tell for an individual what the proportional change in their mutant huntingtin has been over time. So in theory, that might eliminate some of the concern about correcting for Q length, but that still really only works in an experimental situation where we're going to save up the samples, the specimens, run them all at the end within the same assay. Might not work for many longitudinal trials that have maybe a variety of goals in mind, and there are, again, concerns about the stability of assay over time as it's saved. So if we don't do that, we're still back to some of these challenges with assay calibration. So I don't have good answers for any of that, but those are hopefully a little more clearly defined questions, things that we need to grapple with if we really want to get an accurate quantification of mutant huntingtin. With that, I'm going to turn it over to Dr. Southwell.
Okay, so there are some challenges to absolute quantitation of mutant huntingtin, and maybe with the tools that we have now, we can't really do that. And I'm still reporting everything as relative quantitation because I feel like it's where we're at. But even with the tools and the capabilities that we have, it's clear that whatever the CSF mutant huntingtin signal is, it changes. It goes up with disease in individuals, it goes down after ASO treatment. And we want to have a better idea of what those changes mean. And to understand what those changes mean, we need to know where this CSF mutant huntingtin comes from and how it gets into CSF. So we know from previous work that if we use an acute neurotoxin to kill a bunch of neurons, we get a transient increase of CSF mutant huntingtin protein while neurons are actually dying. When the lesion is fully formed and the neurons are no longer dying, this returns to basal levels. So it's an actively dynamic system, and neuron death contributes to it. But neuron death isn't necessary for there to be mutant huntingtin protein in CSF. When we look in the YAC128C6R mice, who, unlike the parent YAC128 mice, do not lose striatal neurons and do not show elevated plasma NFL, suggesting that they don't have neurodegeneration, they do have ample mutant huntingtin in their CSF, as much or more as the YAC128 mice. So, we know that killing neurons causes a release of CSF mutant huntingtin, but it's not necessary for neurons to die for it to be there. So we've looked at active transport mechanisms, and a transport mechanism, a secretion mechanism, was identified for Huntington, an unconventional lysosomal late endosomal secretion mechanism. And we've studied this in primary neurons from our humanized mice, either looking at them untreated or treated with an inhibitor of this pathway. And when we treat neurons with an inhibitor of this pathway, the extracellular mutant huntingtin, this is wild type huntingtin, the extracellular huntingtin component is dramatically reduced. So that shows us that there is secretion of huntingtin from neurons. And we see this from HD neurons, where they secrete both mutant and wild type huntingtin, but importantly, we also see this from normal healthy neurons secreting wild type huntingtin. So we think this is just a normal process. And we went on to investigate this for CSF by giving mice an inhibitor of this secretion pathway. And we saw that a secretion inhibitor reduced the CSF mutant huntingtin component significantly. When we combine this with that acute neurotoxin, we see that even in neurotoxic conditions, inhibiting secretion of huntingtin reduces CSF mutant huntingtin. And conversely, even in the presence of a secretion inhibitor, an acute neurotoxin increases CSF mutant huntingtin. So these things are independently occurring, which suggests that there is both a neurodegenerative component and an active clearance secretion component to the mutant huntingtin in the CSF. So we know that CSF mutant huntingtin increases with disease, and that's seen with our IPFCM assay, and that's also been seen with the complementary Singulex Arena assay, which is now the Quanterix. And if we do a direct comparison of raw signal read for these two assays in different aliquots of the same collections of human CSF, we see a strong correlation. So these things are measuring similar pools of mutant huntingtin. So we know it goes up with disease. So could this mean that there's more cell death? Well, certainly there is more cell death as disease increases, so there would be more mutant huntingtin released passively. There could be changes in the secretion when cells are unhealthy. We don't yet know if disease affects secretion. And there's also this somatic instability that Doug brought up. So there's going to be longer queue as disease progresses. So is it really more protein in the CSF, or is it more assay signal? And right now, it's very difficult for us to tell the difference. So this is particularly important because we know that the majority of CSF mutant huntingtin comes from neural tissue in the brain. And we know that instability is particularly high in the striatum, where cell death is particularly high. So recently, we've used our humanized mice to determine that even with a stable CAG tract, so these mice don't have, these are, the mutant huntingtin in these mice comes from the BAC-HD transgene, which is an interrupted repeat. It's stable, they don't have intergenerational instability, and they don't have somatic expansion. But we do see more protein, more mutant huntingtin in their CSF with disease. So this suggests that the increase in signal with disease is at least in part due to an increase in actual concentration of protein, and not just a change in signal strength dependent on somatic expansion. And so this would suggest that the increase is related to neurodegeneration. But what about when it goes down with treatment? What is this telling us? So we want to know about the brain, but this is an instance where the drug is going into the CSF and the biomarker is being measured in the CSF. And we want to know if that tells us about the brain and about what parts of the brain. And the reason this is important is because in man, these Huntington-lowering agents are not CNS-wide. ASOs have this gradient where the superficial areas have a lot more drug than the deeper structures, whereas virally delivered agents will have much more drug in the deeper structures. So we want to know what we're learning about these various regions. And one of the ways that we're studying this is, again, using these HU9718 mice because they have the BAC-HD transgene and it is floxed. So by crossing to Cree mice, we're able to genetically inactivate that mutant Huntington transgene. And we've crossed them to RGS9 Cree mice, which causes a reduction of the mutant Huntington in the striatum. And this is a very specific reduction. We only see this change in the striatum, not in the other brain regions that we've looked at. And it should actually be specific for striatal neurons, though we haven't confirmed that yet. And importantly, in these animals, we see that when we have this approximately 50% reduction in striatal mutant Huntington, which is within the target range of therapeutic Huntington lowering, we see a consistent and measurable reduction in CSF mutant Huntington. Now, this is stronger in older mice than it is in the younger mice, and this suggests that the striatal component may be biased towards the neurodegenerative component. So this is good news for therapies. You know, it suggests that we can actually learn about the structures in the brain that we're interested in by measuring CSF mutant Huntington. So in summary, as Steve told us, the CSF mutant Huntington assay signal is going to be dependent both on mutant Huntington concentration and Q length. And as Doug told us, Q length cannot be easily corrected for. So this is going to complicate absolute quantitation. But with the tools we have, we are able to tell some things. And we've been able to determine that both neurodegeneration and active secretion contribute to CSF mutant Huntington, that the concentration of protein and not just the assay signal increases with disease, and importantly, that mutant Huntington that's produced in the striatum is detected in the CSF in sufficient quantities to suggest it would be an informative biomarker for striatal therapies. So lots of people contributed to this work, and I'm going to skip thanking everybody by name because we only have a couple minutes left for questions. So I will invite my my colleagues back. You guys have been looking over the questions. Okay. So I think there's some question about how we're able to use this assay in a clinical setting. And one of the things I must acknowledge is that this is not the specific assay that is used in the clinical setting. In the clinical setting, it is the Quantrix assay that is used, but we have done comparisons of them and seen that there is that very nice correlation between the two, and we think that we're able to learn a lot about the biomarker by using this assay. And I think that we'll be able to learn a lot more about the biology of Huntington disease. In future studies, we're going to be looking at these things like whether or not there is a change in the secretion of Huntington from neurons when they're healthy versus when they're diseased, or whether or not within brains, secretion is a neuronal phenomenon. In cell culture, we've seen that neurons secrete Huntington, but astrocytes don't, and we're currently looking into that in brains. So there's a question about, since somatic expansion is inconsistent, what does this mean for interaction among neurons and also for disease progression? I don't know if you guys have a way to address that.
Well, I don't know how to address it, but one thing I've thought about with this, but again, it's something I don't think we really know the answer to, is that if the rate of somatic expansion change is relatively slow compared to, say, a clinical trial, then hopefully it's not much of an issue. But Amber has brought up the possibility that things really are changing dynamically in terms of somatic expansion on a similar time scale. And if they are, then we really have a quandary. But I think that's the basic question about somatic expansion in the brain that we need to answer before we can answer that question.
So I think we're supposed to wrap up, but I just wanted to address one more question from Ed Wild, wanting to know thoughts about the approach of depleting mutant huntingtin and measuring total huntingtin to estimate wild-type huntingtin. And I don't think that is, unfortunately, there just aren't really fantastic ways to measure just wild-type huntingtin. There aren't parts of the wild-type huntingtin protein that it has that the mutant protein doesn't have. So we don't have wild-type specific antibodies. So people have done, independent assays where they measure one or the other and then compare them, but then you're using different antibody pairs for them. And so if you're depleting the mutant huntingtin, your total huntingtin assay signal is still going to be dependent on what kind of antibody pair you're using. And I still don't think you're going to be able to get an absolute quantitation of wild-type huntingtin that way.
Yeah. Total huntingtin assays, we'd have to start over looking at the properties of a totally different assay, and it's a lot more complicated in practice than it sounds in theory.
Okay, we're over time, so I guess we should wrap up.